And so Mark was going to give the introduction, but Mark isn't there right now. I didn't just realize that. Well, that's
I
really
think Mark also went to get a cup of coffee because we said that we were going to chat for a couple of minutes.
well. That's excellent. So we're starting this thing off exactly the way it ought to be started.
And Paul is probably in the background smoking a cigar, which is another. There he
Oh,
is.
not yet.
Sorry, you said we had
Just
a couple.
wondering whether did Mark vote in Florida or not?
I have.
So the
now it's being
recording,
recorded too.
the recording is live. So Mark, why don't you take us
Okay.
away?
So I have not lived in Florida in 23 years, for the record. So it's my honor to introduce today. And again, in advance, I apologize for my garish pronunciation of all words, including names. Dr. Valentina Emiliani, who is currently at the Vision Institute at the Sorbonne. And she's been previously at CNRS. She did her undergraduate work and at La Sapienza in Rome, as well as her postdoc, if I'm reading her bio sketch correctly. This work was in optical investigation of quantum structures. And she moved on to do postdoctoral work also in optics and some lovely work with laser tweezers, which our good friend David Greer probably is very interested in. But then in, I think, 2008 or so, she transitioned to working on a really hard problem in neuroscience. And so this is gonna be a very CSMR specific introduction to this problem, because other people in the world experience this much differently. But if you're in the CSMR, you know optogenetics as the thing we do where we put a bunch of magnets on a plate and we turn on the red light and the neuron is activated and it just works. And that's a combination of happy coincidences that we have very clean driver lines, that the animal is clear, that doesn't see the stimulus and so on. For almost everybody else, you're confronted with the problem that if you want to activate a neuron that's been labeled with an optogenetic channel, there's a lot of other neurons around it that have also been labeled that you don't want to activate. So you need to target the illumination very precisely. You also might need to do it deep in tissue where scattering becomes a real problem. And so the combination of these things, you would naturally say, well, let's just do it with a 2-photon excitation, the same way we do imaging. And it turns out that just doesn't work. People tried this for a little while and it just didn't work. And the reason, if I can be technical, is if you think of the neuron as a Ziploc bag and it's full of goop, for imaging, you just need to get the spot inside the goop. Whereas to activate, all the channels are on the bag. And so you have to hit this very thin bag. And it's not enough just to hit part of the bag because there aren't enough channels there. So you have to hit all of the bag, or a good portion of it, in order to activate it. So you need some kind of oddly shaped light beam. So then you'd say, well, we know David Greer, let's use some kind of SLM or some kind of diffraction grating. But then that introduces a host of problems because when you put a ultra short pulse in diffraction grating, it spreads both in space and in time. And the result is you get MUH. And so Dr. Emiliani developed this ingenious solution to use the fact that the beam is spread in space and in time to force the beam to go through the sample, spread out, and then come together only at the focal plane that you want to excite. And so that is her best, or I wouldn't say best known, but her most cited work is this beautiful Nature Methods paper showing how you can activate neurons using 2-photon microscopy in a targeted patterned way using 2-photon excitation, SLM, and what's called temporal focusing. And now, more recently, you have this problem that this temporal focusing trick, as she initially described it, only illuminates one plane. So if you want to do things in many different planes, then you have, you're kind of back to square one. And so there's this more recent beautiful work using 2 SLMs that allows you then to arbitrarily focus in 3 dimensions light for excitation. And I'm hoping we'll hear some about that today. And I should mention that that work that demonstrates that near and dear to my heart was done partially in dissected Drosophila larvae. So, and it's beautiful. And also in zebrafish with Claire Weir, who we all know as a friend of the center. And so without further ado, please take it away.
Thank you. Thank you, Mark. Actually, basically, you gave half of my talk, so I could skip a certain number of slides. Also, I would really like to thank David for inviting me to give this presentation today. It's really an honor for me to be invited by the father of computer-generated holography. And many thanks to all of you for being here and to be concentrated in science despite the difficult time you are having today. Okay, so I will start now with my lecture, and so the goal of this lecture, as you see, is a pretty broad story that I wanted to tell you today, with the idea of somehow giving an overview on what photonics have done in the past year to provide the technology to control, investigate, manipulate neuronal circuits using light. And before starting to present and to show the different technological advances that have been done in the past year for neuronal circuit manipulation, I choose to start my talk by presenting you 4 important breakthroughs that have been done in neuroscience in the past years to somehow help you in understanding which are the most relevant spatial temporal scales that we need to reach in order to have full control of the brain, or at least to start understanding how the complexity of neuronal circuits work. So the very first breakthrough I would like to mention is the investigation from Ramon Cajal that in the years 1890 or so started with the formulation of the neuron doctrine. And it was among the very first people in highlighting the importance of looking at the activity of a single neuron and highlighting how this is the functional unit of the nervous system. And therefore, with his study, he's also underlined the importance of investigating the brain, having single cell resolution. So in a few years afterward, there was another important breakthrough, and these have been the study from Donald Hebb, which can be summarized by the famous sentence, neurons that fire together, wire together. So in this case, his theory was to put the accent on the importance of neuronal assemblies by providing the picture that actually is the coordinated action of multiple cells that represent the building block of brain function. And therefore, this also highlight the importance that it's true that we need to have the capability to record what happens at the single cell level, but also the capability to simultaneously record from multiple cells to be able to catch the presence of those cell assemblies. There have been another important breakthrough I would like to mention. We are in the year 1950, and this is when Wider pamphlet started to, in this case, by performing electrophysiological stimulation on the larger region of the human brain, he began to map the localization of brain function. And so he drew the first sensory cortex homunculus. And this kind of investigation was of particular importance because he showed how it's important to really have a picture of how brain control behavior, to have the possibility to look simultaneously at multiple brain area. And so this underlined the necessity of large-scale imaging. Very last important step to discuss is about the temporal resolution. And for that, I choose to speak about the recording in 1939 from Hodding and Huxley that for the first time recorded an action potential. So this is the very fundamental electrical signal that neurons use to communicate each other, and is something that happens in few milliseconds. And so therefore, if we want really to see, to manipulate to what happens at the level of signal propagation in the brain, we need to have the capability for millisecond temporal resolution. So all of this is to tell you that we need to have the capability to understand, to manipulate the circuits in the brain. we therefore need to develop a number of technologies that ideally could match all these scales. So single cell to highlight and to investigate what happens at the level of a single element, but also capability to record from multiple cells to start investigate short range circuits, and then mesoscale capability to be able to detect simultaneously from multiple brain area, and finally, as I was mentioning, millisecond temporal resolution to follow the spiking activity at the level of a single cell. Okay, interestingly, optical microscopy can address all these scales, and as a matter of fact, in the past year, photonics has made a very large contribution to neuroscience, with the development of sophisticated optical approaches that in turn have, of course, also inspired new biological questions. And so today, those two disciplines, photonics and neuroscience, are somehow progressing together in a discipline that is called now neurophotonics, which of course is also taking the success from parallel fields like including electrophysiology, biochemistry, molecular biology, and computational approaches. So in this lecture, I would like to focus on the contribution that neurophotonics has made in neuroscience research, and specifically in the contribution that neurophotonics has ported in what we now call the all optical manipulation of neuronal circuits. And when I say all optical, I mean with this the fact that if you really want to understand to manipulate a circuit, as you will see in the rest of this lecture, you need actually the capability at the same time to image what happens in a circuit and to manipulate what happens in a circuit. And so this is what in the modern language we refer now very often as reading and writing a neuronal circuit, or reading and playing a neuronal circuit. So I will now talk about those two parts separately. So let me shortly overview what do we need to read into the brain, and then in the rest of the talk we will focus on what we need for write into a neuronal circuit. So reading the brain, obviously we need, first of all, if you want to do this with optical microscopy, we need first of all to be able to read activity from a single elements embedded in a large three-dimensional networks, and this requires at first the capability to convert this electrical signal I was talking to you about before into a fluorescent signal. And so these so that this can be detected with optical microscopy. And this is something that has been solved with the development of what we call today genetically encoded calcium voltage indicator. So these are fluorescent probes that enable to convert an electrical signal or a change in membrane potential into an optical signals. And you can see an example here where the genetically encoded calcium indicator, as you can see in this film here, enables to transform an electrical signal, which is here in red, into an optical signal in yellow. And so we do have the capability today with optical microscopy to image the propagation of an electrical signal in the brain, in the circuit. And now what about if you want to detect the signal when your cell that you are interested in is embedded in a highly scattering tissue? So for that, we need to be able to send the light, to focus the light on the target, and to go through a scattering tissue by keeping the capability for single cell resolution. So those two challenges, going deep and keep optical sectioning, has been somehow solved by the discovery back in 1930 of the two-photon absorption process. Discovery made by Maria Goppenmeier, which predicted back in 1930 that it appears that a non-zero probability exists that two photons whose frequency sum equals the excitation frequency of the atom coact to excite the atom. So with this sentence, this sentence was predicted the fact that an atom or a molecule can be excited either with the absorption of a single photon, but also with the coaction of two photons having the half of the energy. And so the implication of discovery in microscopy are two. The first is that because we use a longer wavelength in a two-photon process, this allows to penetrate more because the light penetration, the scattering is inversely related to wavelength. And the second important implication is that because for two-photon absorption, we need the coaction of two photons, these give to the effect an intrinsic nonlinearity. And so we can actually have efficient two-photon absorption only when we have enough high density of exciting photons. And therefore, with respect to the single photon process, we have a very efficient rejection of the autofocus light. So only targets which are right at the focus of your beam actually are able to absorb into photon. And so the discovery of the two-photon absorption also marked the beginning of two-photon microscopy, that then was first demonstrated in 1990 by the lab of Wattweb and recognized by many kind of different prizes, among them in 2015, the Brain Prize given to the main actor of two-photon microscopy in biology. Okay, so today, with combining two-photon microscopy together with genetic activity reporter, it is possible to look at what happened in the brain. And here there are two examples of that. So the first one is here. So this is a image is taken by the in vivo from the visual cortex of mice. And you can see that we can actually detect the activity using calcium indicator while projecting different visual pattern to the animal. And what this film is interesting to look at is the fact that we are looking now at genetically identical cells. And despite of that, you can see that each of these cells has a specific role and a completely independent activity pattern with respect to the other. And here there is another example. This now we are in the mouse hippocampal slice. And again, two-photon image using a calcium indicator. And now what you can see is that indeed you have cell with an isolated activity, but also you have a situation where you have a coordinated action of a large population of cells. So those two films that I've chosen is to show you that it's indeed very important to have the capability for single cell resolution, but also the capability to record the same times from multiple cells. So to, for example, in this case, reveal the activity, the coordinated activity of multiple cells. Okay, so from the reading side, using a two-photon microscopy and fluorescent calcium indicators, we can image and see what happens in a circuit under physiological condition. But now the big challenge of these last years have been to try to increase as much as possible the number of targets from which we can record activity. And the limiting factor for that is the fact that the two-photon microscopy in its conventional way is a scanning approach. So a scanning approach means that you can record your image by scanning a spot through your plane. And so as long as you want to record activity from a single plane, for example, for a 500 by 500 micrometer area, this will take between 30 to 100 milliseconds, which is fine to record the activity from this specific region. But now if you want to record what happens on a large neuronal circuits, you need to multiply this number by the several planes from which you want to record. And so you will easily run out of necessary temporal resolution. So in the last year, there have been a number of labs that have find different solutions so to increase the illumination volume in two-photon microscopy. And here I will just quickly go through the different solutions that has been proposed. So one way to increase the temporal resolution when you want to record from a large area would be, for example, to scan, instead to scan a diffraction limited point, to scan what we call the Bessel beam, so that you can, I think that somebody has the microphone on, so and so by scanning with a Bessel beam, is it possible to simultaneously record from a large volume? And more or less with the same idea is to instead of to scan just a small spot, is to create a sort of a sheet of light, and then to scan a line or an entire plane. So this will also increase the resolution. So with this kind of approaches, we today have access to millimeter cubic volume with a 1 to 10 hertz frame rate. We can also increase the volume, the imaging volume, by going for a number of multiplexing solution, which, for example, consist in simultaneously scanning multiple planes or multiple targets. And again, this increase the capability of the number of cells from which you can record. So here there is an example from the group of Ali Fasha Vaziri, where you can see they could record from thousands of cells in a millimeter cubic volume at 5 hertz. There are also a number of solutions to go deeper, so even deeper than what the scattering allows you. And so the first number of approaches consist in using adaptive optics. So this consists in compensate for the deformation that the tissue is introducing into the sample, and so to clean up the extension spot. We can go even deeper today by using 3-photon microscopy. So now, instead of having the absorption of 2 photons, we can absorb 3 of them, so even longer wavelength, so even longer penetration depth. And then if you want to go even deeper, each of this mentioned approach can be combined with a very thin micro objective. So we speak about microendoscopy, and this allows to penetrate even more, and so to reach a brain region which can be several millimeters in depth. So what about the large scale? This is also something that is now under development. So we speak today about mesoscopy. So there are few labs that now have developed the complementary approach, so that we can increase the volume, actually increase the area from which we can record. So this is a spectacular film from the lab of Karl Svoboda, showing that actually today we can do 2-photon mesoscopy, that in this case can record from the entire visual cortex. And of course, you can still have the capability of zooming in with cellular resolution in the region of interest. But we indeed are on the way to be able to record from a multiple area. Okay, so this quick overview was somehow to tell you that on the reading side, there have been spectacular progress in the past years, so that we are really on the way to record from thousands of cells in large volume with cellular resolution and millisecond temporal precision. So now the next challenge is to be able, once we can detect with such a precision what happened in the brain, to be somehow able to replay back what we are seeing, and with the idea to see how this is really affecting behavior, and how modification in the activity that we can see, temporal spatial modification of the activity can modify the induced behavior. And so this enabled me to introduce the second part of the talk, so the writing part. So what do we need to write in the brain? So what do we need to control the activity of neuronal circuits? So of course, if we want to do this with light, the very first step that we need to solve is to making neuronal cells sensitive to light, so that we can ideally use light to evoke or to inhibit an action potential. And this challenge has been solved by the revolution of optogenetics that has been already mentioned by Mark in the introduction. So in this case, it has been shown in the past year that it is possible to genetically express in neuronal cells photosensitive pump or photosensitive channel or pump, so that by using light of different wavelength, is it possible to evoke positive or negative currents through the neuronal membranes. And so this, in the language of neuronal membranes, will correspond to a depolarization of hyperpolarization of the neuronal cells, which therefore give rise to the a cascade of events that can either produce action potential or to inhibit a pre-existing signal. And then we can, in the case of action potential generation, we can also generate a series of illumination pulses so that we can not only generate an action potential, but we can evoke an action potential train, and we can modulate the spiking frequency. So we do have full control of neuronal activity by using light. So you can imagine, obviously, that this is something which is really transforming recent neuroscience research. And again, this is something that has received multiple prizes, and among them, again, the Brain Prize in 2013, given to the main pioneer of optogenetics. So what can we do with optogenetics today? So the main point is that, as I was mentioning in these slides, we can control neuronal activity. And so the very first and most appealing application of optogenetics was to somehow follow what David Palmfield has started many years ago, so to use optogenetics to map brain functioning. And the fact that this can be done by light, of course, has the advantage of being far less invasive. And most importantly, we have the genetic targeting now. So when you send light on a large brain region, there will be only a specific population of cells, which is the one expressing this specific opsin that will respond to light activation. And so even using relatively simple illumination method that simply consisted in shining visible light on large brain region, it was possible to have a very precise mapping of brain functioning with cell type specificity. Okay, so this is super interesting, but now, sorry?
One question quickly is, how do you actually get like genetically modified cells to go in a specific part of the brain? I mean, how do you actually do this?
So, absolutely, good question. I skipped this part. So actually the most common way to have a specific cell type expressing one of those opsins is to use a virus injection, and then the virus is built up with a promoter that recognizes a specific cell type. And so then you normally, this is you inject your virus in a specific brain area according to a specific coordinate, and once the virus is inserted to the brain, he's going to infect selectively only the cells that are recognized by the specific promoter. Then, of course, there are promoters that can infect many cell types, and there are other we can be much more specific. Okay, this is why we speak about genetic targeting, in the sense that you can manipulate your targeting in such a way that there is ideally only one specific cell type that becomes sensitive to light. Okay, so when this is combined with wide field single light illumination, this is already very good to at least correlate what you observe with the action of a specific cell type. But there is somehow a limit in this kind of approach, and the limit is that, of course, when you only use a wide field visible light illumination, all the cells that are sensitive to light will respond in a synchronous way to your light stimulation. And if you remember the example, one of the example I was showing you before, if you actually look at what happened in a circuits under physiological condition, in most of the case, you will see, as in this other example here, that even genetically identical cells might have a very divergent activity pattern. And so the next challenge now is to be able to reproduce this kind of activity. So if we might want now to really understand how those circuits work. And for example, how important is a specific spatial or temporal sequence of signal, and how these can or not control a specific behavior. So if we want to enter in this kind of question, we need also to replace this sort of whole region optogenetics in a more sophisticated combination of approaches that a few years ago we named circuit optogenetics, meaning with this a combination of techniques that, as you will see, can enable you to control the activity of a single or multiple targets independently in space and time. And so let me now guide you through the different technological advances that have to be done to perform circuit optogenetics. And I will also show you a few examples where this has been used to investigate neuronal circuits. So the very first step, of course, to get cellular resolution consisted in replacing wide field illumination with a single cell, with focalized to photon excitation. So exactly as we have seen in imaging, the effect of replacing visible light with two photon excitation in principle give you the capability to get single cell resolution. So unfortunately, as was mentioned by Mark in the introduction, in optogenetics, this was not so straightforward as in imaging, because what at a certain point people realize is that when you use a conventional two-photon microscope that only shine a small spot into a cell body, in most of the case, the number of channels that you can photostimulate is not enough to create enough current that can depolarize the membrane to evoke an action potential. And so people have to find solution in order to perform two-photon optogenetics, solution that could increase the size of the illuminated cell membrane. And today we have two complementary solutions to this problem. So the first way to increase the number of photostimulated channel is to perform a quick scanning of the spot through the cell body. This is an approach that has been pioneered by the group of David Tank back in 2009, that for the first time showed the capability to evoke an action potential in two-photon. And the other complementary approach is to go for a parallel approach, where instead of increasing the portion of illuminated membrane by scanning, you can modulate the optical wavefront, so you can cover the entire cell or the multiple target that you want to excite. And this we demonstrate this the first time back in 2010. So obviously the main difference among those two approaches is in the temporal resolution. So if you go for a scanning approach, the temporal resolution that here here with temporal resolution, I mean the illumination time that you need to evoke a certain response. So in case of a scanning approach, this temporal resolution take into account the time you need to scan through the cell, the time to go from one target to another, and of course will depends on the number of targets. So today, if everything is optimized, roughly to activate 50 neurons, you need between 50 and 100 milliseconds. Now, if you go for a full parallel approach, so where the target or the multiple targets are simultaneously illuminated, then the only time that matters to define the temporal resolution is the illumination time. And this, as you will see afterwards, today can be as short as 2 to 5 milliseconds. So we can reach hundreds of neurons in a 2 to 5 millisecond temporal resolution. Okay, so in my lab, we have been particularly interested in trying to optimize the temporal resolution. And so we have been focused in the past year in the developing of optical approach that could deliver light in a parallel way. And specifically, we have been concentrated in two complementary approaches, computer-generated holography and generalized phase contrast method. And both of them make use of a liquid crystal device. So is a device that allows, as you will see, to generate arbitrary light pattern at the sample plane. So for a lack of time, I won't have time to go through all of them. And so I will, from now on, concentrate on the approach of computer-generated holography, which is also the one which is more mostly used in optogenetics today. So computer-generated holography is based on a very old principle in physics. Holography, which I guess many of you knows already, is a revolutionary invention that made by Dennis Gabor back in the 1970s. And that idea was to develop something that could recreate the shape of physical object with light. So the system is actually very nicely shown here. So you need a laser. You need to split the laser in two beams, one that we can call the reference beam, and the second one illuminating the object that you want to eventually recreate with light. And then this next step is consisted in recording the interferogram between the reference and the diffracted beam. And the last step was to illuminate with the reference beam your interferogram, and this will create a virtual and a real image of the object. So if you look through the hologram, you will have the perception of the object. If you put somewhere a camera, you could record the shape of the object recreated with your reference beam. So the idea of holography has been largely exploited in science fiction, where people were speculating the possibility of creating three-dimensional animated holograms. And actually, this is not so far from what we can do today. And this is what makes the difference between a static, the recording a static hologram, and what we call today computer-generated holography. And so the main difference is that we don't need anymore to have the physical presence of the object. This can be simply an image template that you draw by computer. You don't need to record the interferogram. This can be calculated with a Fourier-based iterative algorithm. And most important, you don't need a static phase hologram, but this phase hologram can be addressed on a liquid crystal matrix, so that you can illuminate the matrix with your reference beam and recreate the shape of the object at the objective focal plane, and having the advantage of dynamically refresh the holograms. So the idea of computer-generated holography was first exploited from the community of optical tweezers. And so here the holograms have been used to generate multiple diffraction limited spot that then were used to trap, to tweeze micrometer size beads. And so there have been the group of Hest and David Greer have been the pioneer in the field of computer-generated holography. And this is actually a film from the group of David showing how it was possible in computer-generated holography to simultaneously control the location and of multiple beads in 3D, creating those three-dimensional holographic light distribution. Okay, so we can generate a multiple diffraction limited point, and this was used in optical tweezer, but we can also generate a more complex shaped pattern. And a few years ago, we actually showed that this can be done in a very precise way. So to the point that you can start by recording a fluorescence picture and then define the part of the region that you want to illuminate back with your light, and use this as your image template, so to produce a phase hologram. And if things are well done, at the sample plane with your laser beam, you can reproduce a pattern that precisely reproduces the region that you have selected. So this can be, for example, a dendritic region, the dendritic process, or can be a specific group of cells that you have selected from a larger population of cells. Again, as it is in the case of optical tweezer, you can send multiple holograms, so your shaded light can also be animated in time. And so this is just an example where we projected an hologram into a fluorescent cover slip to show that we can generate animated light shapes. Okay, so the very first application of holographic light for neuronal control was using single photon light and cage compounds. So this is an alternative way with respect to optogenetics to control neuron activity with light, and this consisted in introducing in your preparation what we defined, for example, in this case, cage glutamate. And so the cage is a photosensitive cage that can be broken by sending light. So this will correspond to a release, in this case, of glutamate. And so this will generate a current in the close body cells. And so we first proposed this in single photon, and in the same year, the group of Rafael using multiple diffraction limited point in 2D showed two photon holographic uncaging, and then mine and other groups has also extended this approach in 3D. So we have to, for optogenetics, we have to wait few years more. But now I think for what I told you, you will immediately understand the interest of holographic light shaping with for optogenetics, because this was really the solution to make your extension spot bigger, eventually of exactly the size of the target that you want to illuminate. And of course, then you can generate a illumination shape that either illuminate a single target or a certain number of cells which are close to each other. So there is a problem when you, as you can imagine, when you increase the illumination size, and the very first drawback of that is that you immediately lose axial resolution. You can see this clearly here. Okay, so this is the axial propagation of the beam that at the sample plane will create this configuration here. So as long as we look at the axial propagation of the part of the beam creating a single spot, this is pretty good. We have between 10 and 40 micrometer, depending on the spot size. So we are on the size of a single cell. But now, if you illuminate multiple cells at the same time, you will see that the axial resolution quickly deteriorates. So we lose optical confinement, even if we are using two-photon excitation. So we need somehow to find a solution for that. So if we wanted to use shaded light, we need to find a solution to having back optical sectioning. And the solution that we proposed was to combine two-photon holography with the technique of temporal focusing. So, which I'm going to explain you now. So temporal focusing is a very elegant approach that has been first proposed by the group of Silberberg and Kritsu back in 2005 to perform two-photon wide field microscopy. Okay, so let me go through the principle. So for that, you need to remember what I was mentioning at the beginning, and the fact that the key properties of two-photon absorption is that we have a quadratic dependence on photon density due to the fact that in two-photon microscopy, we need the coaction of two photons, okay? So because of this nonlinearity, if you use a strongly focalized beam, you will have a very high density of photons at the focal plane, while the density of photons get lower when you move out of focus, and this gives you the optical sectioning characteristic of two-photon microscopy. But now we have seen before, for what I told you before, this very small illumination volume is not enough to evoke a current using optogenetics, for which we would rather like to illuminate the entire cell membrane. Okay, so if we do so, we can't have a focalized beam, so we will lose the optical sectioning characteristic of two-photon microscopy. So there is, however, a way to recuperate the optical sectioning if you can increase the extension density instead of using spatial focusing, by using a sort of temporal focusing. So by using a sort of time lens that temporarily concentrate the photon at the focal plane and temporarily dilute the photons out of focus. Okay, so what is this time lens? Actually, this time lens is nothing more than a dispersive grating. So this is the basic scheme for temporal focusing. So basically, you need a pulsed laser. This is what you would normally use into photon microscopy. And then what you do is to send your laser through a dispersive grating. And so the effect of the dispersion is that the different colors of your laser are going to be dispersed through slightly different angles. And this will introduce a difference in the optical path between the different color. And this difference will be larger the more we go away from the grading. Okay, so this difference in the optical path will introduce a broadening of the pulse duration. And so now if your grading is conjugated with the sample plane, you will have a short pulse at the focal plane, while moving out of the focal plane, your pulse will become broader and broader. And so now, because the two-photon absorption is inversely proportional to the pulse duration, this will give you an intrinsic optical sectioning even without having a focalized beam. So in our specific case, we wanted to also keep the flexibility of shaping light in 2D. And so before reaching the grading for temporal focusing, we go through a spatial layer modulator, which is creating a specific shape on the grading, and then the grading takes care of temporal focusing. And so now if you look again at the propagation that I was showing you before, you will see that in the presence of temporal focusing, we can nicely concentrate absorption around the focal plane, and so get an axial confinement between 9 and 10 micrometers. This will, of course, depends on how you build up your system. But what is interesting to mention is that now this number is independent of lateral spot size. So no matter which is the illumination shape that you want to realize at the focal plane, you will always have an axial confinement and always of the same size. So the combination of lateral eye shaping and temporal focusing enable to increase the efficiency into photon optogenetics and keep axial resolution, as we first demonstrated with Francesca many years ago. And here are so examples. So here we demonstrate actually that we can generate multiple, we can control action potential generation using two photons, simultaneously multiple cells or multiple targets, and having axial confinement. So there have been multiple variants. Actually, in this specific case, the approach for lateral eye shaping was generalized phase contrast, and we build up variants using holography, single photon and two into photon. One I would like to show is that this experiment. Was particularly challenged because was using holography and calcium imaging in single photon now, but in freely moving animals. So in this case, the holographic system was coupled with a fiber bundle, and then we use the holography we use the imaging path to look at different neurons, and then the holography to select the target or the targets that we want to activate. And the advantage of having a fiber bundle was that we were able to control, as you can see here, the activity of a single cell in a freely moving animal. So this was done in single photon, but despite of that, we could get almost cellular resolution because, as you can see from the fluorescence image here, we use a sparse staining. So we have very, so the stimulation spot were very far away. To get true single cell resolution, as I was mentioning before, we need, of course, to use two photons and this combination of lateral and axial shaping. And actually, speaking about cellular resolution, as people realized afterwards that a very precise light shaping alone is not enough to get a true single cell resolution, and for that, actually, to get a true single cell resolution required the help of many labs working on molecular biology, and that today are able to provide what we called soma-restricted opsin. And the need of that is shown in this cartoon. So what actually at a certain point became clear is that when you have a dense distribution of cells expressing a specific opsin, even if you can be very precise in where you shine your illumination volume, still you might have many processes from the surrounding cells that cross your illumination volume. And so therefore, even if the light is only here, you might have multiple targets going to be excited. And this will obviously completely deteriorate your spatial resolution. And so many labs in the past year have found a solution to that by developing some opsin that can restrict the expression of the opsin only in the soma. And so by doing that, we can today reach almost through single cell resolution. There is an example of that here. These are recording that we did in our lab from the visual cortex layer 2-3 in vivo. And you see, we can, for example, now define that these 3 cells have to spike. And so we send 3 spots there. And as you can see from the calcium traces here, the 3 cells responds. And now we can decide that only one of them is going to be excited. So send the light only there. And you see that actually only that cell is going to respond. So this is to say that we are really, by combining targeted light with soma-targeted opsin, on the way to get a single cell resolution in the manipulation of neuronal circuits. So I will show you an example of where this precision was of key importance. This is an ongoing collaboration with a group of Rosa Cossart in Marseille, in France. So here they are interested in demonstrating what they demonstrated already years ago in vitro, and is the existence of hoop cells. So the idea that there are cells that alone are able to control the activity of an entire circuit. So the experiment was performed in developmental barrel cortex. And at first they performed calcium imaging to follow the activity of the network. And on the base of the calcium activity, they could define highly connected cells. They could do a sort of, let's say, functional connectivity analysis to find the highly connected cells. And then the experiment was the following: to use holographic light patterning and the soma-targeted opsin to activate those highly connected cells. And as is shown in this movie here, so you start by recording the activity of the network. You see this is a highly active network corresponding to a specific inter-event interval. And then you start photostimulating just one cell, one of these hoop cells, and you see that the activity, the activation of just one single cell is able to inhibit, or at least to slow down significantly the activity of the network. And then when the photostimulation is off, the activity of the network goes back to the baseline. And so this was a very important result, because showed that actually with the control of the activity of just one single cell, is it possible to control the activity of many cells, to actually drive the activity of the entire network. And in this specific case, this was a specific properties of highly connected GABA neurons. Okay, so we do have the capability to control neuronal activity with single cell precision. And the next challenge was now to be able to get this precision in the case where we want to control multiple neurons at the same time. And so for that, the first challenge was to be able to scale up the number of neurons that we can reach with this kind of manipulation. So now if you want to increase the number of targets, there is basically no choice. You need to go for a parallel approach. You need to reach them with an holographic approach. And you have 2 ways of doing that. You can either use a spatial layer modulator to create a multiple diffraction limited point, exactly as was done with in an optical tweezer system. And then you can scan those spots in a spiral way, so to get to have multiple neurons excited simultaneously in 3D. And this is an approach that has been first demonstrated by the group of Michael Heuser a few years ago. Or again, if you really want to optimize the temporal resolution, you can go for a full parallel illumination approach, which means holographic shaded light combined with temporal focusing to get axial confinement. Obviously, the fact of having at the same time temporal focusing and 3-dimensional light shaped required to find a solution to have temporal focusing and 3D light shaping. And the solution we proposed was to, as was mentioned by Mark in the introduction, to use now 2 spatial layer modulators. So a first one for the control of lateral edge shaping, and the second one for the control of the axial position. And of course, in between, you have the grating for temporal focusing. So this configuration has then evolved. And so because I won't have time to go through the whole story, let me just show you which is the configuration which is mostly adopted today. So as I was mentioning before, we have a first spatial modulator, and this is creating a shape on the grating. So in most of the case, when we want to excite multiple neurons, you don't need to have independent shape. You can just create, for example, a disk of the right size, and then project this on the grating for temporal focusing. And now the second SLM now is used exactly as we use in holographic optical tweezer to generate multiple diffraction limited spots. And now when the two of them are working together, you will have a convolution of these two distributions. So basically, what the second SLM does is to create a multiple replica of all what the first SLM is doing on the grating, and all of them are going to be temporally focused. So there are multiple variants of this approach. So you, depending on how you decide to produce this first shape on the grating. So this can be, for example, replaced by a simple Gaussian beam, or can be a general phase contrast method, or can be a mixture of amplitude phase modulation approaches. I won't have time to go through all of them, but you can see them reviewed and compared in this review here. So here, let me just show you one example, and is the example where we have twice holography. And so this is shown here. So the first spatial modulator is creating an holographic disk, which is temporally focused. And the second one, as is shown here, is now creating 50 replica of the exact same disk in 3D, 50 different axial position. And because each of these is just the exact same replica of the same spot, they are all temporally focused. And so with this, you can get an axial resolution between 10 and 12 micrometer in the entire field of view, and which is the number you would need to get a cellular resolution when this is combined with a soma-targeted opsin. Okay, so what's for? Why do we need to generate three-dimensional pattern of spot? So there are two applications I want to show you where this has been used. The first one is to make advantage of this capability of three-dimensional light shaping to decouple the imaging from the photostimulation plane, so that you can perform a sort of all optical investigation of multi-layer circuits. And this is what we used in an ongoing collaboration with a group of Serge Picot, Olivier Mar, and Denise Dalcara here at the Vision Institute. So in this experiment, now we are in the mice retina, and the idea was to investigate how the output from the bipolar cell layer into the ganglion cell layer is distributed. And so for that, you should know that ganglion cell layer and bipolar cell layer, they are separated by 50, 70 micrometers. And so if you want to create a specific activity pattern into the bipolar cell layer while looking at the calcium response evoked into the ganglion cell layer, you need somehow to decouple the imaging from the photostimulation plane. And so this is something that we could do thanks to the fact of having holography into the photostimulation pattern. So the microscope was combining a conventional two-photon microscope to image the calcium response in the ganglion cell layer, two-photon temporally focused holographic system to project a specific activation pattern into the bipolar cell layer. And then we also have a visual simulation to, as is shown here, to be able to characterize the different ganglional cell type according to how they respond to a specific stimulation protocol. And so then the experiment was that the first question was how local is the output generated from the bipolar cell layer into the ganglion cell layer? And what turned out is that actually this is pretty local in the sense that we can have evoke a response into the ganglion cell layer as long as the group of cells that activate the bipolar cell layer are between 50 and 100 micrometer from the targeted ganglion cell layer. And the second results was, and so this was demonstrating that the rod bipolar cell output is locally integrated in the ganglion cell layer, so demonstrating that there is a limited spatial divergence. And because of that, in the case of direction selectivity of cell, the asymmetry is preserved, meaning that depending where the activation pattern in the bipolar cell layer is produced with respect to the preferential direction of the direction selectivity ganglion cells, this generate a strongly asymmetric response in the ganglion cell layer. Okay, of course, to explain really the results, we will need a separate talk, but here the message I want to pass is that in some case, the capability of shaping light in 3D is that this open the possibility also to investigate the interaction among multiple circuits, even when they lie on different axial plane. Then there is a second obvious interest in developing approach for three-dimensional light patterning is that we might want to control entire circuit, or at least we might want to answer to a key question today in neuroscience, and this is given certain circuit, how many neurons I really need to activate to have an effect into behavior. So for that, I have selected three very nice examples that has been published in the last 2 years. So the first two came from the lab of Carl Deisseroth and Raphael Just. So here the experiment was the following. They wanted somehow to see how many cells we need to control in the visual cortex to bias perception. And so the experiment was consisted in 2 steps. The first step was to perform 2 photon calcium imaging to identify activity patterns that correspond to a specific visual stimulus in a go-no-go behavioral task. And then the second step was to reactivate those patterns using a 3-dimensional holography and see whether by light it was possible to induce a specific behavior. And interestingly, the results was that this was possible. So actually the main conclusion was that yes, with the control of just a very tiny fraction of neurons is enough to bias, in this case, the visual, otherwise visually induced behavior. Another very impressive paper that recently came out, this from the lab of Shai Shoam and Dima Rimberg here at New York University. This is, now we are in the olfactory bulb. The question was somehow similar. So it was to which extent the mice can feel the photostimulation. So how many neurons we need to activate to give the perception of the stimuli. And there have been 2 important results in this paper. The first to actually demonstrate that in most of the cases, less than 20 neurons are enough to evoke a response in behaving animals. And the second important result was to show that it's not only the number of neurons that matter to give the perception, but it's also the temporal synchronization of the stimuli. So actually they compare the response when these multiple neurons are simultaneously excited within 10 milliseconds, and then they start to desynchronize the stimuli so to spread the stimuli to 30 to 50 milliseconds, and this was enough to lose the perception of the stimulation. And so this is really important results, because it really showed that at a certain point is not only the number or the spatial location of the stimuli that matters to control behavior, but also the temporal distribution of the stimuli. And so this enabled me to also somehow introduce the very last part of the talk, and this is about the time. So I really till now concentrate on the importance of get cellular resolution and the importance of control activation of many neurons. And now what is something that is very important and is to start to be able to investigate how much the synchronicity of a certain number of stimuli can or cannot control, or let's say how much the synchronicity of a certain number of stimuli matter or not to control behavior. And so somehow a very dream experiment that we start to have using all these approaches together is the one that I show in this cartoon here. So at a certain point, we might want to reach a situation where we can detect activity from a circuit and then be able to exactly reproduce the same activity using light, using optogenetics. And so for that, we need to multiple things. First of all, we need to be very precise. So we need to be sure that when I send the light here, I can precisely predict when this neuron is going to spike. And then I want to probably to reproduce slow spiking and fast spiking cells, so that we need to have a temporal solution so that we can generate action potential train at very high frequency. And if we want to project multiple patterns with a certain temporal sequence, we need to do this in a very fast way. Okay, so for temporal precision, the good news that actually if you use parallel illumination, so that you can very efficiently integrate carriers into, you have a very efficient carrier integration. This allows you to use very short illumination pulse, so typically between 2 to 5 milliseconds. And so because the illumination time can be very short, this also enables to have a very sub-millisecond temporal jittering. So actually, if you repeat the same stimulation several times, we and other labs show that you can reproduce the same spiking time within a sub-millisecond temporal resolution. And so having this precision, you can register, for example, as this was done, these are data from my lab in vivo, you can record the spontaneous activity, and this again, layer 2-3 of the visual cortex, and it precisely produce exactly the same activity within sub-millisecond temporal precision. About the spiking rate, what it turned out to be very important is the developing of opsin having a fast photocycles, and so that you can be sure that after the first pulse, the opsin is quickly going back to the equilibrium and is ready to be excited again. And so if you combine opsin with a fast photocycles together with parallel illumination, we can actually control action potential generation at very high spiking rate, 80 to up to 100 Hz, using, for example, chrome of crimson air. So we do have also the capability to generate a high spiking rate. So the very last point I was mentioning before was for sequential light. If you want to perform fast sequential patterning, and today, if you want to generate multiple illumination pattern, the limiting factor is the refreshing rate of the liquid crystal method. Okay, so you can't switch from one hologram to the other with a time shorter than 3 to 20 milliseconds. So what about if we want to go faster than that? What about if we want to really investigate how much desynchronization shorter than that matter in controlling behavior? And so to go faster than that, we propose this last variance of holography, which is shown here, where now before reaching the second spatial lane modulator, we have a galvanometric mirror. And then the spatial lane modulator can be now addressed with multiple holograms. And what we can do then is that the light temporally focused, which becomes a line, is illuminating these holograms. And then with the galvanometric mirror, we can quickly move the line through the different holograms. So now imagine that each of these holograms is encoding for a different spot or for a different distribution of spot. By going quickly through all of them, you can generate, as is shown here, a sequence of patterning that in principle at the kilohertz frame rate. And so with this, we can control, for example, the spike delay between two cells with the precision of the microseconds, and also to control independent spiking rate from multiple cells or from multiple group of cells with the same kind of precision. Okay, so I think this is time to conclude now. And I hope you are still there. And the conclusion is a general conclusion that I would like to make now, is that there have been really complementary fields that participate to this long story. So photonics is important, but opsin engineering was important, laser development, modeling, and of course also neuroscientists that are able to understand what the technology can do, and therefore to ask the right question that we can answer with this technology. But altogether, all these people bring today the field of optogenetics really in a new phase, where we can think of start playing with the circuit. And although there is still a certain path to do for that, we can really start thinking of try to ask how specific spatial temporal activity patterns are linked to animal behavior. So just before the very end, let me thank first of all the people of my lab, the people that are currently in my lab, and the different people that contribute to the different data that I show you in this long lecture. And the many collaborators, again, because this is a highly interdisciplinary field, we have collaborators both in the opsin engineering part, in the discussion of waveform shaping approach, and a long list of neuroscientists that help us in developing the right tool for the right question, and then the technical support also from Coherent 3i and Amplitude. I would like to thank, of course, the different financing source, and of course, a big thank for your attention. Thank you.
Mark, are you going to lead us?
Sure. So that was really beautiful. Can I ask a question about the last bit with the multiple spot switching?
Yeah.
It seems there must be some trade-off in resolution if you can't use the whole SLM.
Yeah. I didn't have time to go through that. Of course, this is something we characterize. So, and what we, of course, the more holograms you add, the very first things you can lose is light, in the sense that, of course, your hologram has to be equal or slightly larger than your temporally focused line. Mm-hmm. And because, I mean, it's smaller than that, you will lose light. And you can, if you reduce too much the number of pixels, you can have a sort of elongation of your spot. And then at a certain point, you will start losing also in the resolution. So what we have seen is that actually, as long as you keep the number of holograms in a single run to be about 20, so 20 holograms for the conventional Hamamatsu device, this is a good compromise. I mean, you don't have any significant drawback. More than that, we require either to focalize more the line or to have a compromise in the resolution.
So
but actually, yeah.
So you up your frame rate by 20, essentially.
I would say, yeah.
Impressive. Can you use it for imaging?
Yeah. This is also something that could be interesting. Yes, to have a fast X, Y, or zeta scanning. Yeah.
So okay, so you're getting the speed up higher and higher. Is there any sign that the neurons just can't keep up? I mean, what's the fastest you can ping neurons before they stop paying attention?
So actually for that, let's say the speed is not really, let's say you will, the limiting time to evoke a response, you still need to illuminate your cell for a certain time. Let's say between with existing opsin, when the expression level is good, this can be as short as 1 millisecond, but let's say you still need 1 to 3 milliseconds to evoke a spike. Okay.
And that's limited by the gate opening or by the ion gate opening, or is it limited? I mean, what limits the sort of
the gate opening is pretty fast. Is on the order of a few femtoseconds. I mean, what really required to illuminate your target for a certain time is the number of carriers that had to flow through to generate enough current to evoke an action potential. You can say it's the two photon cross section somehow. Okay. And then also the conductivity of your channel. So you need to stay for a second. You need a total illumination time of 1 to 3 milliseconds to have enough current flowing through. But as you said, actually, it's not really the opening time. So actually, you can either illuminate continuously of 3 milliseconds or just going back several times, so that at the end of the story, what is important that this total illumination time generate enough carriers. Okay. So the capability, the interest of switching very fast is, imagine, as is shown in this example, imagine now you have those two cells, okay? And you want to generate an action potential in those two cells. So if you illuminate them simultaneously, they will both spike simultaneously, okay? Then they could spike not exactly simultaneously if the expression level is different, but assuming that the expression level is exactly the same, you send two spots, they spike at the same time. And now imagine you want to start to see how important is the synchronicity, or if you want to desynchronize those two cells. So today, this was limited by the fact that if you want to desynchronize them, you have to stimulate this one and then that one. And so to go to there, you are limited by the fresh rate of the lipid crystal. With our approach, we can go much faster. But still, the limiting factor is the illumination time. But now imagine that you have 3 holograms, okay? This first hologram send light there, this second hologram send the light there, and this one send the light in both cells. So now if you have this, you have actually the liberty of doing whatever you want, because you can keep the illumination of this first hologram as short as you want, and then this is going to be your spike delay, right? So the speed, so this is to say that the problem of the speed is not really that the neuron can follow. For us, the speed was important to reduce this spike delay as short as we wanted. I don't know if I was clear.
I get it. Absolutely. It's
Okay.
really cool.
What about energy balance? You're using so you actually haven't talked about the pulse energy, because the regular laser, pulse laser, would probably enough to stimulate all these hologram and multiple spots. So you're using the amplified lasers, right?
Yeah. What's your question, Dima? Sorry.
And that sets a limit how. So you're using amplified lasers, right? So basically the point is that when you want to stimulate multiple holograms, the regular pulse energy is not enough for 2-photon stimulation, what my understanding is. So you need to have much more.
Your question is, why do you use pulse amplified laser? Is this the question?
What's the limitations there? Because amplified laser has a much lower frequency. Which frequency are you using? And this
but no, we are never going as fast as the. I mean, this switching time is on the order of the microseconds. So the pulse. Normally we work at 500 hertz. So that's not the limiting factor yet. I mean,
500 hertz, you have enough energy to stimulate multiple neurons. That's a little, because basically what we were dealing is, if you're taking 800, 500 hertz laser, the pulse energy is not large enough. So we use the amplified laser and significantly reduce the frequency. To be able to feel so basically that have, I mean, sorry, I didn't want to distract. Are we already in a free discussion, or we still, you know, I can talk about it a little bit later. But basically, with a regular laser, energy of the pulse is not enough, especially if you need to fill the whole neuron with light.
Sorry, Dima, what do you mean with a regular laser? Is the T-Sapphire laser, the 18 megahertz?
Yes.
Okay. Yeah, so for what you call the regular laser, 80 megahertz laser, we do have energy to excite a single cell. In our experience, depending on the opsin and expression level, I think one can realistically excite 5 to 10 cells, okay?
What is the volume of the cell? How large is the cell, and how long do you need to keep the light on the cell with 80 megahertz laser?
But the illumination time is more or less the same, 2 to 3 milliseconds. So 2 to 3 milliseconds, 5 milliseconds, you get a cell to spike.
And what's the volume of the cell?
But the volume, the cell is always the same, right? So this.
Well, some cells smaller, sometimes larger. So if you scale it up,
well, let's say for a pyramidal cells, layer 3, T-Sapphire laser, we could excite, let's say, from 5 to probably 10 cells if everything is at the optimum. Okay.
Now, 2 milliseconds. Wow, that's very, very. So basically, we've been struggling with that because we don't have enough energy for 2 milliseconds. We need to shine the laser for long enough time on the cell in order to excite, because it's basically not enough energy, not enough 2 photons.
Okay, but as you said, this depends on the cells. You're thinking about the micro cell.
And Valentina,
any more questions, and then come back after we stop
Sure.
recording?
I didn't want to.
Sort of like a last chance for people to get in more sort of the general questions before we dig into the details. But maybe people are shy. So maybe what we should do is stop recording so that we can dig into the details. So let's do that.
So I'll say thank you again.
Thank you.